Okay, hello everyone. So welcome to biology lesson with me, teacher Hanisa. Okay, in here we're going to discuss about the last chapter in the Form 5 syllabus, which is genetic engineering. So actually we have two subtopics for this chapter, but in this video we're just going to look at 13.1 is about the genetic engineering. So here is the learning standards for today's video. We're going to describe the terms of what is genetic engineering and also genetically modified organisms, or also called as GMOs. Alright, and then in the end of this lesson, you are able to explain the application of genetic engineering in productions of the GMO, genetically modified organisms, and also genetically modified foods. And the last one, how these genetic engineering is involved in the productions of insulins. All right. So we're going to look at the first one, the definitions of genetic engineering. So genetic engineering, also called as genetic modifications, genetic manipulations, it is a direct manipulations of organisms genes or organisms DNA using a biotechnology, right? So they manipulate, they modified, they change the organisms DNA genetic using a biotechnology. So that is a genetic engineering. All right. So an organisms that is generated through genetic engineering is considered to be genetically modified. All right. And the resulting entity or this resulting organism, also known as genetically modified organisms, GMO. Okay, so now you know genetic engineering is an application, but genetically modified organisms is the results from the genetic engineering. Okay, so next, this is examples of genetic engineering. So you can see in the slide, we have a regular fruit. So this is an apple. So this apple, we modified this genetic of this apple. So we insert the copy of insect resistance, meaning that we want to change this regular fruit, this regular apple. So these fruits can have a trait or characteristics of insect resistance. All right, so what we do is, this is what is genetic modification or genetic engineering. We insert this copy of gene for insect resistance, and then we're going to the results from this practice, or the results of this genetic modification will gives us a new fruit, but this time it's not a regular fruit, it is a insect resistant fruit. So this is the GMO. All right, so we're going at what is GMO, genetically modified organisms. So basically, genetically modified organisms, GMO, is organisms that contain recombinant DNA. All right, and organisms that contain recombinant DNA is also known as transgenic organisms. All right, so meaning that the genetic has been changed, right? So there has two names here. You can call it as a genetically modified organisms or also transgenic organism. It's the same thing. So what is recombinant DNA? Recombinant DNA, so DNA is recombined. There's a new set of DNA, segment of DNA is being inserted into the DNA of that person. So this is the vector, and then this is the DNA fragment. So the DNA fragment means this is the desired characteristic that a person want to put in these particular organisms. All right, so this is a recombinant DNA, and then this recombinant DNA being put in the organisms. All right. So now you can see here, there's examples of genetically modified organisms. We can see this is cow and this is a goat. Kambing dan juga lembu. So apa yang genetik dia ubah? This is genetically modified cow because it produces milk which does not contain beta lactoglobulin, a type of protein that causes allergy among some children. So basically, the normal one, the cow milk, it contains that beta lactoglobulin, and some children cannot drink it because they're allergic to this protein. But scientists, it has been they've already modified the cow, so will produce milk that doesn't contain this beta lactoglobulin. All right, so when that happen, many children can get benefit from the milk. Okay, so the next one is another examples of genetically modified organisms, which is goat. Okay, goat. This genetically modified goat, it possess the human gene, which causes for blood clotting factor. Okay, so this blood coagulation factor is found in the goat's milk and can be purified and used to treat hemophilic patient. Okay, patient. So hemophilic, what is hemophilic? You know, hemophilic is a disease disorder where a person with hemophilic traits, their blood cannot clot easily. Okay, dia tidak darah. Seorang yang hemofilik ni, dia mempunyai masalah darah lambat membeku. So dengan ini, apa yang saintis buat, saintis masukkan, sebab orang yang hemofilik ni dia kurang faktor pembeku. Okay, faktor pembeku tu dimasukkan kepada kambing. So kambing ni akan menghasilkan susu kambing. Dalam susu kambing tu dia ada faktor pembeku tu. Okay, blood clotting factor. So bila seseorang yang ada hemofilik minum susu kambing tu, dia akan dapat manfaat blood clotting factor tu. Saudara dia akan boleh membantu orang yang hemofilik ni supaya apabila diorang mengalami pendarahan, darah diorang akan cepat membeku. Alright? So that is a GM. Okay, I forgot to change it. This is supposed to be GMF, right? My bad. So genetically modified food, now GMF. Alright. So genetically modified food, tadi organism. So kalau organisms basically dia lebih general. Organisms ni boleh plant, boleh tumbuhan or anything. Tapi ini food ni dia spesifik untuk source of food. Maksudnya, saintis dia memang pilih trait tu untuk menambah baik karakter ataupun traits, ciri-ciri makanan. Makanan ni of course maksudnya plants dan juga tumbuhan yang memang untuk dimakan. Okay, so genetically modified food (GMF) possess DNA from other species of plants or animals, and consumptions of GMF by humans may cause health implications which are still unknown. So dia masih ada debate around this topic, macam this issue genetically modified food. Ada yang menentanglah this teknologi, mungkin ada efek dia apabila kita ubah genetik tu. Okay, so this is how we identify, how we know that food is genetically modified. Okay, so basically the characteristic of genetically modified food is the food, the plants or animals has a resistant to herbicide. Kalau herbicide ni plants lah, the resistant to herbicides, lebih tahan daripada tumbuhan perosak, resistant to disease, tahan daripada penyakit, applications in medicine, and also tolerance to heavy metal and resistance to pest. Okay, ini adalah ciri-ciri makanan yang telah diubah secara genetik. Dia lebih tahan. Okay. But there are advantages and disadvantages of the genetically modified food. Of course, kenapa orang start buat benda ni? Kenapa orang buat genetically modified food? So, kita nampak dia punya benefit dulu. So, what is the benefit? Of course, benefit ni, genetically modified ni, maksudnya bila saintis dia ubah suatu genetik, suatu plants tu, sebagai contoh tadi, kita ambil contoh in the earlier slides I shows you the apple. This regular apple tu dia mudah rosak, dia tidak tidak tahan lama. Tapi bila modified, the genetic is being modified, dia lebih resistant, insect resistance. Okay, maksudnya dia lebih tahan lama, jadi kita boleh manipulate dan kita boleh hantar makanan ni boleh digunakan oleh ramai orang. Dia tidak cepat rosak. Maksudnya kalau tahan lama, kita boleh pasarkan ke tempat lain, ke tempat yang jauh and all that. So advantages, we're going to look at the advantages first. So it can overcome worldwide food shortage by producing high quality transgenic crops and livestock. Okay. Macam tadi itulah worldwide. Bila kita ada kualiti yang bagus, dia boleh tahan lama, kita boleh hantar pergi negara-negara yang kurang sumber makanan. Okay, dan kita tahu kalau hantar makanan tempat, contohnya daripada kita tanam di Malaysia, kita tanam dan kita hasilkan buah itu, and then kita hantar pergi negara Afrika contohnya. Malaysia dengan Afrika kan jauh, so dia mengambil masa yang lama. Jadi untuk mengelakkan buah tu rosak dan tahan lama, inilah kita gunakan teknologi BMF. Alright, and also reduce cost of food productions. Okay, dia mengambil cost yang yang mengurangkan cost, and then increase nutritional value of crops and reduce problems of crops related to pests. Macam tadi itulah, kita boleh pilih traits yang kita mahu. Kita mahu tumbuhan tu dia lebih tahan daripada, dia tidak boleh, dia boleh menghalang haiwan perosak and all that. Okay, reduce usage of pesticide. Jadi kalau secara genetik kita boleh halang daripada haiwan perosak, jadi kita boleh mengurangkan penggunaan pesticide. Okay, racun perosak lah. Racun untuk elak haiwan perosak. Okay, and also the last one, it will increase the production and reduce price of food. Thus, increase food availability. Okay, kalau kita dapat tahan lama, kita dapat hasilkan makanan yang tahan lama, and then kita boleh hasilkan, bila tahan lama, stok akan lebih banyak. Okay, stok lebih banyak, jadi itulah maksudnya increase food availability. Okay, dan bila ada stok yang banyak, kita boleh turunkan harga makanan tu. Dia tidak terlalu mahal sebab makanan tu available sangat banyak. Jadi dia bukannya benda yang rare. Jadi so itu kita boleh turunkan, reduce the price of the food. Okay. Tapi there are the disadvantages of this genetically modified food. The first one is endangered and natural species. Maksudnya yang semula jadi itu terancam. Kita sudah ubah genetik dia. And then there is a slight possibility that the foreign gene in GMF may be transferred to humans. For example, antibiotic resistant gene. Bahayalah sebab antibiotic resistant gene ni dia akan ada possibility bila kita makan GMF tu, kita makan makanan, we eat the food that already being genetically modified, so it will affect our body. Okay, kalau genetik, kalau maksudnya antibiotic resistance ni, badan kita dia akan melawan antibiotik dalam badan, macam dia ada efek. There is a slight possibility, ada possibility. Alright. And then it may have adverse effect on human health and genetic material. So kalau kamu perasan perkataan di sini, slight possibility, and also it may have, dia bukanlah confirm. So dia masih lagi undergo this research still is undergoing research, has been doing this, still in ongoing research happen, okay, tentang kesan GMF ni, okay, dan disadvantages. Tapi dipercayai takut bila kita mengubah sesuatu tu dia akan ada efek yang tidak baguslah kepada kesihatan manusia. Okay, so that is the advantages and disadvantages of genetically modified food. Kalau kamu boleh pergi baca artikel-artikel ataupun boleh baca bahan-bahan artikel tentang research GMF ni, memang ada pro and contra lah. Memang ada sesetengah community yang tidak bernyokong tentang GMF ini. Okay. Next, we're going to look at examples of genetically modified food. So the first one, for example, I take these examples in your textbook. The first one is how corn is being produced using this recombinant DNA technology. Okay, whenever I said recombinant DNA technology, it also mean genetically modified food. Okay, kan ingat tadi ada recombinant DNA, maksudnya DNA yang telah recombine, diubah-ubah. Okay, so what is the new gene that has been inserted in this corn? So there's a Bacillus thuringiensis gene is inserted into corn to increase its resistance against pests. So maksudnya corn ini, jagung ini dia lebih tahan daripada hama. Okay, and then there's another examples. You can see here in the picture. This is from your textbook. You can see the comparison. The same. This is salmon. The same species. Both are salmon. Dan sama usia adalah. Okay, dua-dua ni adalah 8 bulan. 8 months. This is a normal salmon. This is a super salmon. Kenapa dia panggil super? Sebab yang super salmon is genetically modified organisms. It's a genetically modified fish sudah. Okay. Sebab apa yang diorang buat ialah it is modified by inserting a growth hormone gene. Growth hormone ni maksudnya hormon yang hormone that catalyze the growth of organisms. Okay. Growth hormone is being inserted in growth hormone gene is being inserted in the super salmon. Okay, dari mana dia ambil growth hormone gene ni? Daripada Chinook salmon. Lepas tu dimasukkan kepada genome of Atlantic salmon. Okay, apa maksud genome? Genome ni adalah maksudnya a set of DNA. Maksudnya dia satu set. Lepas tu kena dia kasih masuk dalam satu set DNA ni, kita ada masukkan yang baru lagi. Itulah insert into a genome. Okay, genome of Atlantic salmon. So super salmon is created. So this is a new, dia bukan a new species, cuma dia a new, what we call it as a new recombinations of DNA. Okay, so this super salmon is created. It will grow at a faster rate and can be produced throughout the year. Sepanjang tahun dia boleh membiak. Okay, next. Okay, next is another examples of GMF, genetically modified food, but this time is about potatoes. Okay, potatoes, kalau negara barat lah, okay, di western, in the west, dorang tidak makan nasi ternasi macam kita kan. Jadi salah satu sumber carbohydrate is from the potatoes. So potatoes is important crop to people who lives on the cold climates. So it is very sensitive to low temperature and frost. Maksudnya potatoes ni memang makanan ruji, makanan utama, the main course for the main food for this person, for the people who lives in the cold climates. Tapi potato ni pula dia sensitif pada suhu yang sejuk. Jadi untuk menggalakkan potatoes ni kekal boleh bertahan pada suhu yang sejuk, what they do, they modify the genetic of these potatoes. They put a gene from Arabidopsis species plant, which enables the plant to tolerate freezing condition. Okay, dia masukkan genes ni masuk ke dalam potato genome. Again, genome ni apa? Genome ni is a set of DNA. Okay, and then kenapa dia insert? So that you would create a genetically modified potato yang masih boleh tumbuh. It still can grow well in a cold climates. Okay, so sebenarnya GMF ni dia memang ada kebaikan dia jugalah. Dia ambil ciri-ciri mana yang diperlukan dalam satu organism. Contoh macam for examples, for now in this slide, potatoes. We need potatoes to continually grow even though it's in a cold climate. Okay, dalam suhu yang sejuk. So, they modified. Diorang ambil genetic genetic that brings a desired characteristic atau traits, masukkan ke dalam inilah genetic, the genome of the potatoes. Cara diorang masukkan semua tu memang secara makmal lah. Okay. Alright, now we'll look at insulin. Tadi kita sudah tengok GMO, genetic modified organisms. Lepas tu kita sudah tengok genetically modified foods. Sekarang kita tengok insulin. Okay. Insulin ni pun di bawah genetic engineering juga. Okay, kenapa? Sebelum tu kita tahu insulin function dia adalah dia digunakan dalam kita punya hemostasis. Dia kontrol kandungan gula dalam darah. Okey. Dan tetapi insulin ini sebenarnya insulin ini dia boleh, selain daripada it can be produced naturally in our body, it also can be produced artificially. Boleh dihasilkan sendirilah. Maksudnya sendiri, boleh dihasilkan di makmal. Okay. In the older days, insulin was extracted from the pancreas of cattle or pigs to treat diabetes mellitus patients. Okay. Seperti kita tahu, diabetes mellitus patient ni maksudnya patients with diabetes mellitus, there's a high concentrations of glucose in their body where insulin cannot cooperate with that. Insulin tidak boleh tukar sudah. Dia punya glucose itu become glycogen. Jadi, insulin daripada luar need to be injected into the body. Dari mana dapatnya insulin dari luar tu? Dari dulu diambil daripada lembu atau pig khinzir lah. Okay, extracted, dikeluarkan. Nowadays, insulin can be commercially produced by genetic engineering. Maksudnya commercially produced ni maksudnya sudah dikilangkan, sudah dijual. Okay. And then, besides insulin, other successes of genetic engineering includes hepatitis V vaccine, blood clotting factor, and also growth hormone pun boleh dihasilkan secara makmal. Okay, di luar. Mindset inilah macam commercially produced. Kita sudah beli. Kita sudah boleh beli di luar. Kita tidak payah lagi extract daripada mana-mana haiwan yang lain. Okay. Alright, so sekarang kita akan tengok macam mana insulin itu dihasilkan. Okay, sebenarnya dia ada satu picture yang besar, tapi I break it down into two supaya kita boleh tengok satu-satu. Okay, so situasi dia macam ni. This is insulin gene. Okay, insulin gene. Memang hebatlah. Sebenarnya macam mana orang tahu itu adalah insulin gene? Ini adalah secara memang sudah di-identify genome. Kita sudah tahu set DNA kita tu mana satu yang DNA, mana satu yang bawa karakteristik untuk warna kulit, untuk warna mata and all that, yang simple karakter yang kita nampak lah, okay. So memang tahu sudah locationnya di mana. So this is insulin gene, a split human DNA with insulin gene. Okay, so insulin gene is cut with restriction enzymes. So macam mana ini adalah insulin DNA. Okay, sorry, ini adalah human DNA yang ada insulin. So kita ambil insulin DNA, cara kita ambil dia, kita cut dia menggunakan enzim. Dia bukan ambil dia kan macam tu, dia guna enzim. So enzim tu dia akan hydrolyze ikatannya. So kita dapatlah keluarkan extract insulin gene. At the same time, pada masa yang sama kita ada satu plasmid. Apa itu plasmid? Plasmid is a circular DNA. Okay, plasmid yang membentuk dia mesti bulat. Dia plasmid ni dia daripada bakteria. Okay, bakteria which is used as a cloning vector. So cloning vector maksudnya dia vector ni kalau bahasa ringkasnya lah dia pembawa. Okey, maksudnya kita mau kasih masuk insulin gene itu, kita tidak boleh terus direct ambil insulin gene terus kita kasih masuk dalam DNA, tidak boleh. Dia mesti ada pembawa dia. Pembawa dia inilah plasmid ni. Okey. So plasmid ni sama juga kita akan cut dia gunakan restriction enzyme. Kenapa kita cut? Supaya on the third step, kamu tengok di sini. Dia ada recombinant plasmid. Maksudnya, yang tadi plasmid tu vector kan? Plasmid is a vector. Pembawa. So maksudnya ini adalah, sorry mana sudah camera. Okay, so ini adalah plasmid. So ini adalah gene. Insulin gene yang kita sekarang kita menghasilkan insulin kan, ini adalah insulin gene insulin. So kita cut plasmid tu supaya kita boleh masukkan insulin gene dalam plasmid tu. So apa yang terhasil kita boleh tengok dalam slide ni, a recombinant plasmid which is successfully inserted with human insulin gene. Okay, dia masukkan. Alright, so DNA ligase enzyme. DNA ligase enzyme ni maksudnya yang inilah dia ligase, dia memotong. Okay. Next, step yang keempat. Okay, step yang keempat, the recombinant plasmid is introduced into a bacterium. Okay, plasmid tu kita buat, itu plasmid. Sekarang ada satu lagi bakteria. Bakteria ni kita masukkan plasmid tu tadi kan, plasmid ni sebenarnya berasal daripada bakteria. Kita kasih masuk dalam bakteria. Kenapa masuk dalam bakteria? Bakteria ni kita boleh biakkan dia. Sekarang ni kita mau banyakkan, kita mau cloning, kita mau kasih banyak itu insulin. Okay, maksudnya kita ambil satu gene insulin, kita kasih masuk dalam bakteria, bakteria tu kita kasih banyak. Jadi kamu boleh nampakkan daripada satu gene insulin dia jadi banyak. Okay, so kamu akan nampak sini, the recombinant plasmid is introduced into a bacterium. So this is a transgenic bacterium. Kenapa kita panggil transgenic bacterium? Sebab dia bawa satu plasmid yang sudah ada genetik yang baru, iaitu insulin, human insulin gene. Alright. So bila dia dia masuk transgenic ni, kemudian number 5, bacterial clone multiplies and produce insulin. Daripada satu dia akan kena clone. Dia akan dibanyakkan. Alright. Jadi lepas tu nanti insulin is extracted from the transgenic bacteria and purified. Sudah banyak nanti barulah insulin ini dikeluarkan. So maksudnya daripada satu boleh hasilkan banyak. So insulin ini akan dikeluarkan semula daripada bakteria itu. So kita ambil insulin inilah dan akan digunakan secara komersial. Siapa yang perlu insulin, dia boleh inject dalam badan dia. Alright. So ini adalah overview apa yang kita bincang tadi. Daripada step pertama, dia kena ada left and right. Saya memula insulin, ambil daripada gene human insulin gene extracted. Pada masa yang sama, plasmid pun extracted from bacteria. So nanti dia akan combine human gene masuk dalam plasmid. So plasmid yang ada human gene tu akan dimasukkan dalam bakteria. So bakteria ni akan culture, dia akan dibiarkan jadi banyak, multiplies ataupun being clone banyak. Okay, dan sudah jadi banyak, kita ambil lagi semula, keluarkan insulin yang banyak tadi tu, and then purified, then boleh digunakan lah. Okay, okay, now the summary. So okay, so summary. So again, kita sudah belajar tentang terms tadi, genetic engineering. Kalau kamu mau ingat balik, genetic engineering dia mesti ada kaitan dengan modifications ataupun manipulations of gene through biotechnology. And then genetically modified organism is something that is being produced. Something apabila kita sudah modified sesuatu genetik itu, dia akan menghasilkan genetically modified organisms. Okay, and then we already look at this application of genetic engineering dalam 3 benda ni: genetically modified organism, genetically modified foods, and also insulin. Insulin kita bincang dalam silibus kamu tu lah yang lebih detail kita mau tengok. Maksudnya bermula daripada apa, insulin ni diambil daripada mana dan dimasukkan dalam mana, plasmid. Okay, and then genetically modified organisms and foods, kamu sudah tengok dia punya advantage dengan disadvantage. Kalau advantage, dia akan kurangkan cost, it will reduce the cost, it will increase the food availability. Ya, dia akan, makanan itu akan jadi banyak. Okay, and then disadvantage tadi ada juga disadvantage. It may slightly have adverse effect to human health. Macam itulah. Okay. So kalau ada apa-apa soalan, nanti kamu boleh tanya teacher. Jadi that's all for our video for today. So thank you very much for watching this video. So if you have any questions related to today's topic, you may ask me in the WhatsApp. Okay, thank you very much. Stay safe, everyone.